Good afternoon. Welcome, everyone. It's just about 3 o'clock, so we wanted to get started. My name is Marie Leteliep, and I am a manager of some of our nutrition programs at ILSI North America. We wanted to welcome you today. So this series is brought to you in collaboration with the American Society for Nutrition, and what we're doing is exhibiting the ongoing work of faculty and students of the University of Illinois, and all of this work focuses on the gut-brain axis and the gut microbiome. So a housekeeping item, just to say that we are recording this webinar and it will be available on our website after the fact. So we'll be sending everyone a note that that's ready. And also in the end, we'll open this up for questions from the audience. So please feel free to use that chat box throughout the talks, and we'll get to those at the close of the session. So with that, I would like to turn the microphone over to Dr. Wendelin Jones for a few opening comments.
Great. Thanks, Marie, and thank you everyone for taking time out to join us today. Want to welcome you to this ILSI North America webinar. For those of you who are not familiar with us, ILSI North America is a nonprofit scientific organization that advances food safety and nutrition science for the benefit of public health. We do not lobby. Rather, as the only US and Canadian organization that brings together scientists from government, industry, and academia, we are focused on solving those challenges that can only be solved through collaboration. With that, on the next slide, want to also highlight how you can get in touch with us and how you can learn more about us. So first thing I want to highlight is our web address, which is ilsina.org. Want to encourage you to follow us on Twitter and want to encourage you to follow us on LinkedIn. We are committed to being transparent in all of our activities, and you can therefore find out much more about us by following us on social media. Additionally, if you have questions, our email addresses are shown on this slide. So lastly, right before I pass it back, I want to thank all of you. I want to thank our presenters from the University of Illinois, and I want to recognize our collaborations with ASN. So with that, back to you, Marie.
Great, thanks, Wendelin. And with that, I'm very pleased to introduce our first speaker, Dr. Kelly Swanson. Dr. Swanson is the Kraft Heinz Company Endowed Professor in Human Nutrition at the University of Illinois. And his laboratory studies the effects of nutritional interventions on health outcomes with a primary emphasis on gastrointestinal health and obesity in dogs, cats, humans, and rodent models. Dr. Swanson is also quite active in ILSI North America, and he serves as an advisor to our Gut Microbiome Committee and also as well as on our leadership group. So Dr. Swanson, we turn the platform over to you.
All right, thank you, Marie. I'd first like to thank ILSI North America and the Division of Nutritional Sciences. I know Dr. De Mejia and Marie in particular put this together, and I think it's a nice webinar series. Hopefully you were able to join last week, and we have again a few more weeks of this. And so if I can figure this out here, I'm gonna bring up a laser pointer that hopefully everyone can see, and I think I'll probably turn off my webcam so you can focus on the slides. It's probably more relevant than what I'm gonna than seeing me in my office here. But again, today we're gonna focus on sex-related differences in host metabolism, health, and then I guess the role of the gastrointestinal microbiota. And I think at this point it's certainly a question, not a statement. I think a lot of people are really interested in this area, and so I think it's something that should be studied and is of course highly complicated. So it's in kind of an unwritten story, I think, at the moment. But I want to share a little bit of what we're doing in our laboratory and what other people are doing with this focus in mind. And so before I get there, let's see here. Okay, here we go. A brief outline of the talk today. I have, again, about 30-35 minutes, and then I'll turn it over to one of my PhD students touching on a little bit sexual dimorphism in humans, whether it's metabolism and disease, the gut microbiota, and then turning over to some of the animal models that we use in our laboratory, and especially looking at the ovarectomy or OVEX model. And then I'll turn it over to Celeste, where she will has done some work that's kind of taken that OVEX model to another level, and I'll let her focus and kind of explain what she's been doing the last few years in the laboratory. Before I get there, though, I want to give a little bit of background of where I'm coming from when you think about studying the gut microbiota and relating that to health. And so this is my kind of a big picture of my laboratory and of my research experience and my expertise. And so I've focused in the GI health area for about 20 years, including my graduate work and now in my laboratory. And a lot of what we do, I am trained as a nutritionist. I'm not a microbiologist, and so I'm often looking at ingredients, dietary formats, and how that impacts not only the gut, but also other things related to obesity and metabolic health. And so been working in this area for quite a while. As the slide also suggests, I use a lot of animal models. We conduct some human research, but we do a lot in the rodent model more recently. And for 20 years, I've been doing research in dogs and cats, and really not as a model for humans. Almost all of it is exclusively with application to the pet food industry or veterinary medicine. And so I'll touch on that. But when we really focus on the gastrointestinal health and microbiome area, it really we kind of use a three-pronged approach. And so I'm a nutritionist, so I really want to know, and really when you think about the microbiome, you need to know the chemical composition of the diet of the ingredients being tested and being studied. And then we often will do some screenings with in vitro testing. And so oftentimes we're focused on dietary components that are carbohydrates, but it's very important to know what components we're talking about. A lot of people have product streams that they're not really sure it's high in carbohydrate, but are they oligosaccharides? Are they digestible carbohydrates? Are they dietary fibers? And so we'll often do in vitro testing to answer this question: is it going to be digested by the host enzymes, or will it be not digestible and it will be potentially fermentable by the microbiota largely in the large intestine? And so if that's the case, then you can start looking at the microbiota, the metabolite, the gas kinetics that come from that as a screening tool to apply to the target species. And in our laboratory, oftentimes it is the pet, but also it's humans. And so these are more applied studies where they're non-invasive, where we're really focused on either GI function and or obesity. And so my foundation really is in fibers and prebiotics. Been working with them since day one, really, of my research career. More recently, we've looked at yeast fermentation products that are probably most accurately categorized as postbiotics, different macronutrient profiles, not only from a metabolic perspective from the host, but also from a fermentation perspective. Really have a huge shift in the microbiota and the metabolites if you shift from protein fermentation to carbohydrate fermentation and vice versa. So we've done a lot of work in that area, as well as whole foods with certain collaborators that I'll mention at the end. Again, these studies are non-invasive, so we can look at gastrointestinal intolerance. The tolerance factor is always important, whether it's yourself or whether it's your pet. The fecal microbes, the metabolites, breath hydrogen, immune cell populations. There's more and more phenotypic variables that we measure, but these are the main ones. So again, I've been doing the work here in the top two bullets for about 20 years. The last 5 to 10 years, we've adapted a little bit and opened ourselves up to kind of using rodent models a little bit more. A lot of the things you can show, either whether it's in vitro or in vivo with humans or non-invasively with pets, you kind of run into a roadblock a little bit. And so we still measure effects of diet, but also look at obesity and metabolic health. I'm going to talk today about ovarectomy. Celeste is going to talk about gallbladder removal. And then, of course, not everything's going to translate to humans. So we have to be careful with that. But certainly you can measure and make a lot of measurements and do things that you can't do in humans or other animals sometimes. And so we've tested gut barrier function, again, immune cell profiling, and from a tissue perspective, looking at tissue, different mRNA gene expression, as well as gut microbe and metabolite concentrations as well. And the key thing here is we're not reliant upon fecal samples at this point. We can use cecal samples or other samplings in the gut that is probably more relevant to the physiological status of that host versus just a fecal sample. And so again, a few things that we do in our laboratory. One thing that got me really interested in, really actually early on, what initially got me interested in kind of these sex differences is actually our work in pets, where you spay or neuter an animal, or your dog or your cat, and they, within a few weeks, and we've done this several times, but in the home, but also in a laboratory, they're going to eat more. They're going to start eating more. Within a few weeks, you'll see changes in appetite. Of course, they're going to gain more weight. Metabolically, they're different. Physical activity is reduced. And so that really got me interested. But another thing is some of our human studies, and this is the best example we've had of this, is that when we start conducting our human studies, I think we have to, people are already considering the sex of the individual. You know, there's a lot of inclusion, exclusion criteria, but we have to, I think, when we look at the response to dietary interventions, and maybe actually how we stratify our subjects, we have to think about microbiota, and some people are already doing this. But certainly this is just an example here that Hannah Holscher was a postdoc in my laboratory. And this is, I'm just giving one example of fecal bifidobacterium populations from agave inulin supplementation. And so we saw a treatment effect. And so what we have here is in this bubble plot is the response versus a control that was 0 grams per day, or the 5 or 7.5 grams per day of agave inulin. This was a Latin square design, so every person on the study, we had 30 that began, we had 29 that finished, so we had 1 male that dropped. But they finished the study, and we were looking at bifidobacterium in particular to see their response to agave inulin. And what you'll see is that the top panel here, you can see minor changes, and about half of them seem to have an increase, a slight increase in bifidobacterium, and the other half here did not. You flip to the females, and you'll see about 2/3 of the females responded, and a few of them responded in a great fashion, actually. So we had an overall treatment effect, but it was largely driven by female. And so my initial thought was it's a dosage effect. They're smaller, they're eating less calories, so you know, per 1,000 kcals or whatever you want to look at it, that dosage was larger because we kept the dosage the same. But it could be that the microbiota populations were different slightly, and we weren't powered really to look at that necessarily. But also, is estrus cycle playing a role? And we tried to try to compensate for that, but there's only so much you can do, and maybe there's other factors as well. But certainly, studies like this and data like this again got me thinking even more. And so I think this is an area that's again, the story is certainly untold at the moment. It's still an evolving story. But I think it's a very interesting one. What's not, new necessarily is the difference between males and females when it comes to metabolism, when it comes to disease risk, and in particular obesity and the comorbidities. And so certainly there's been just a lot of publications. It's well known, the body fat distribution from a male that has an apple shape, if you will, the female has a more of a pear shape, glucose tolerance, insulin signaling, fat deposition outside the adipose tissue, so liver and skeletal muscle, lipid metabolism, inflammation, the list goes on and on, male versus female. The disease risk, again, for the majority of life after puberty, males have a greater risk of obesity and these comorbidities. And so this figure kind of shows this pattern. I'm only going to focus on the developed countries here. The developing countries, they have similar increases again in the level of obesity, but I'm really going to focus here. Really what you'll see again, once puberty has been completed, in an adult state, males have an increased risk of obesity and all these other diseases. And this goes until about the mid-40s. What you'll see here now, and you hear with infants and with animals, sometimes you'll say there's catch-up growth. Well, after menopause, there's certainly some catch-up, but it's a negative catch-up here. We don't want this catch-up. And what women will, on average, again, will do is they'll kind of catch up to men. So by the time you get to the mid-60s, they're almost at the same level of obesity and a lot of these comorbidities. And so when we think about sex, and we really have to think about what, on the female side in particular, what stage of life are we talking about? Because menopause really, what I've said here is flips the script. This is an example on the right panel here, blood pressure. And you could draw a figure like this for many different conditions, metabolic conditions. But what you'll see again, through puberty, basically to menopause, there's this protection effect that females have, and then it quickly changes within a matter of a few years. You can see this blood pressure is going to increase. And so when you think about other cardiovascular disease and some of these comorbidities, again, insulin resistance, properties here, inflammation, there's a lot of changes that occur. Of course, there's a lot of factors involved, but menopause is one of those key factors that needs to really be studied more, I think. And so that's where we're going. Of course, we're talking about microbiome here, and so like everything else, we try to match to see what role does the microbiome play. And so I've just thrown up here titles of several papers in the last couple of years that have tried to tackle this question. And many of these are review papers. Only one of them has a question mark. This one on the bottom here is a question mark. The others are kind of statements, but I think for the most part, it's a question mark. What role does the gut microbes have when you think about host health, but also you throw in the combination of male versus female? And I think one thing to think about it, we're always saying, what is the gut microbe? What are they doing to us? I think we really need to think about it the other way as well. People say the gut microbiome is kind of a super organ, and I agree that it is. And it's probably the most modifiable and adaptable organ that we have in our body. That it might not be the same taxa, it might not be the same gene content, but they certainly adapt to their environment. And so whether it's sex hormones, whether it's the diet that we eat, the substrates they see, or competition with other microbes, the communication they get from gut cells and other signals that they receive, they're adapting to things all the time. And so we really have to think about, I think it goes both ways, and I think the data will kind of demonstrate that. But what people have done, they've looked at cardiovascular disease, again, inflammation, immune system. There's a new term, again, it was new to me. It's only recent here, the microgenderome. And so again, we're combining now the microbes, the hormones, immunity, and disease susceptibility. So there's a lot of things at play here, but again, they're all fairly recent. And so I thought, for this talk, I could throw just tons of microbiota data at you over the next several slides, but I'm not going to do that. I think it's an emerging area. I think we're very early stages, just like we were, I would argue, 10 to 15 years ago, looking at the gut microbes and just pick any disease. Some of the initial studies, or a lot of the initial studies, was healthy versus disease. It's very exploratory. There's a wide variation, a lot of confounders, especially diet, which I'm happy to see is more of a being either controlled or at least monitored more nowadays. But there's still a lot of these studies that don't really even look into diet. But we have, of course, race, medications, any other exposures. So a lot of the initial studies are looking at male versus female, with considering and trying to tease out a lot of these confounders, but it's not quite that simple. You can't mathematically just rule out a lot of things, and so people have tried to do their best, but we need more and more prospective studies, I think, to see what's changing. The Shin paper that I've shown a couple of figures here really took it to another level, and they looked at hormone concentrations, and they compared males versus females, but within females they also looked at the low estradiol group, the high estradiol group, and then they plotted some of these relationships. And so it's interesting to see, okay, here's some taxa here, and they show what phyla, but they get down to the genus level to see what is enriched in one group versus the other. When you start plotting them out, though, you'll see we're at early stages here, that this is nothing that would be diagnostic at all. And if you actually look at what would probably be all the postmenopausal women on this axis right here, where there's basically zero serum estradiol, they scan the gamut when it comes to the relative abundance of these different taxa. It's as wide as it can be. And then there is a line drawn, but I don't think anyone would put too much confidence into that. Nothing against the researchers or the research being done here, but I'm just, we're not there yet. They're still very much exploratory. On the rodent side, again, rodent models are commonly used as well, of course. And again, several people, I've just put a few citations here on the slide, but several people have shown this interplay between sex hormones and the microbiota, and then what impact that has on phenotype of the host. And so estrogen here shown to affect microbial diversity. Gut microbes mediating how estrogen has an effect on disease, whether it's endotoxemia, whether it's inflammation. In gnotobiotic mice, again, these were conventional, but in gnotobiotics, if you keep an animal germ-free, these sex responses, male versus female, are not nearly as dramatic, and so they're very much reduced. And so there's just one citation that's shown that. Again, the microbiota altered the sex hormone response on autoimmune disease, similar to above. And then male and female, very interesting, the recipient mice, these gnotobiotic mice that received donor microbiota, they accepted the microbiota differently. So what that means is that the males, after microbiota were transferred into them, the populations that ended up inhabiting the males were different than the females. And so again, there's something going on here, but it's very, very complicated. And so when you dig down a little bit, I said, my lab really has a three-pronged approach. There's another, even if you dig down even deeper, it's the molecular, basically molecular microbiology. And we have Jason Ridlon is a great example here on our faculty in animal sciences that really studies a lot of this at the molecular level. And he focuses mainly a lot on bile acids, but the sex steroid hormones are very similar to bile acids in a few ways. And so one, before we get to their metabolism, and the recycling in the body that occurs, I'll just kind of say again, we're mainly with males. We're talking about testosterone, and it's relatively stable over time. Again, if you look at the metabolism, there's a lot of hormones and things being transferred from one way to the next. But really, we're looking at testosterone levels in the male. In the female, it's quite a bit more complicated, and so certainly there are several estrogens. We have E1, 2, and 3, and so the primary, the most powerful estrogen is estradiol, E2, synthesized in the ovaries. Once you go through menopause, if you're a woman, certainly estrone becomes the more predominant, and this is synthesized in the adipose tissue largely. And then the placenta for during gestation and pregnancy, estriol, E3, is produced by the placenta. And so there's different forms that play a role here, and they're variable over time. So again, we're going to focus probably on menopause here, but certainly during pregnancy, a lot of people have looked at the microbiota during women and during pregnancy, and then transmitting those, that translation of course, not just genetics, but the inoculation of the microbiota of the infant, but even estrous cycle. When we're thinking about conducting our studies, there's hormonal changes all the time, and this is probably going to affect the microbiota over time as well. So we need to consider that. Where it gets even more complicated, really at the molecular level and enzymatic level, is that in the liver, similar to, again, bile acids, they are derived. A lot of these estrogens, androgens are derived from cholesterol, and they can be modified in the liver in many different ways. And I'm going to focus on conjugation and the recycling of these. And so just like the bile acids are conjugated with amino acids, mainly taurine and glycine, these sex hormones can be sulfated, they can be methylated, and they can have a beta glucuronide unit can be added as well. And so they can be excreted through the bile, and they're in the gastrointestinal system. But then the microbes come along and they can deconjugate. Again, similar to bile acids, they have sulfatases that can break off the sulfate group, the beta glucuronidases, and there are groups of genes. The GUS gene is one we measured in our laboratory with the help of a collaborator that can deconjugate, and then again, like bile acids, can be reabsorbed, go back to the liver, and they'll be used again. And so what's interesting is if you have really strong antibiotics, this recycling decreases at a very large level. In one study, it even was like 60-fold reduction in the amount of recycling that's occurring. And so if you take the microbes away, a lot of this changes. And so certainly there's a lot of things going on here. In my laboratory, given our experience, given our expertise, I guess we rely on the animal models to take a closer look here. And so we're doing not just looking at sex, we're doing the other studies that I mentioned before, where we're trying to look a little bit closer at this with rodent models. Really, again, getting closer to causation and mechanism. Some people say we're gonna demonstrate causation. It's very difficult to do that, and given all the factors that are involved here, you're getting closer, but it's difficult to do that. What I really like, though, is that there's a shorter time, certainly, versus studying humans, of course, and the amount of control you have. And so I think, again, I'm preaching to the choir here with a lot of nutritionists on the line, but we can control diet. We can, and I just I have to say it every time, is get on my soapbox and say we can't make much progress in this area, especially when you're looking at sex hormones and how they're being recycled using chow diets, because there's a lot of natural phytoestrogens that will really bias the studies and lead to responses and data that don't really match what you think you're measuring. And so we really need to use specific semi-purified diets to do that. But there's other exposures you can limit in this situation. Of course, tissue samples, getting closer to mechanisms, germ-free options, and surgical models as well. So there's a lot of benefits there. The challenge is I always have to just recognize here, we have to be careful what translates. And so unfortunately, and Celeste is going to talk about this, a lot of the work in rodent models is done in males. And NIH and other agencies and funding areas of funding have focused on, okay, we need to look at females, not just males, but the great majority is done in males. And there's a reason if you're looking at obesity for this. And over here on the slide, you can see that they gain weight quicker, they develop obesity and all these comorbidities much quicker, and you can avoid a lot of the hormonal cycles that occur. And so in many ways, from a scientific perspective, it's much cleaner, it's easier. But then do the data really apply to the population or just to males? And so that's the question that must be asked. Of course, we have to recognize the physiological differences, the gut microbiota differences between the models and humans. Certainly in mice, if you are looking at gut microbiota, you need to be collecting at the cecum level. You can measure feces, but it's still the cecal tissue and cecal samples is probably where you need to be. The other thing we need to recognize is there are some taxa that are different, and there are some taxa in rodents that aren't even present in humans. And so people get kind of up in arms about that. I think we need it. What it stresses, though, is a lot of the biology and the physiology going on is similar. So it stresses the need to focus on gene function, enzyme activity, not taxa. And I'm guilty of it as well. It's very easy to generate 16S data, and that's fine. But we have to also look at the other the mechanisms here, what genes are involved, because there's so much functional redundancy in the microbiome. Many of these things, it probably doesn't, maybe with some of these mechanisms, matter which bacteria is performing the function. It's that the function is being performed. And so it really depends on what you're looking at, but we have to focus on that. For the rest of the talk, I'm going to kind of focus on some of the work that Zhu-Wen Cross did when she was in my laboratory. She's currently assistant professor at Purdue University and did a postdoc with Federico Rey and it was University of Wisconsin-Madison. And so she's moved through that system. But while she was in my laboratory, conducted several very, very nice experiments, and one that I'm going to focus on was on the effect of ovarectomy. Of course, we had our hypotheses and were able to demonstrate that food intake and energy metabolism, lipid accumulation changed. But we tried to do is explain a little bit more from a gene expression perspective in these tissues that are important, inflammation, as well as the microbiota. And her overall question here was taking this, and this is not even a lean mouse here either, but it looks a little bit chubby, but taking this mouse into a level of obesity. And the model that we use is the C57 Black 6 mice. But really her questions were, when does it change? And is it diet, is it OVEX, or is it both? And so we haven't answered all the questions certainly, but I think we've developed a nice foundation and a lot of interesting data, complicated data, but interesting data on this topic. We didn't get to study this necessarily, this enterohepatic circulation, but we did measure the beta-glucuronidase enzyme, or at least a collaborator of ours did, and so we'll touch on that briefly. But a lot more work needs to be done. Well, again, she conducted many studies, but I'm going to touch on just one of them here. And this was the kind of the primary study, and it really ended up being several chapters of hers, is that we had a study where 40 mice arrived, 10 weeks old. They were able to acclimate and were allotted to dietary treatments when they arrived at the facility. And they had 2 weeks to adapt to that diet and to the facility. At baseline here, we collected fecal samples, and then Zhu-Wen performed the ovarectomies when they were 12 weeks of age. And so what we ended up having was a low-fat fed diet, high-fat fed, and this is about 10% calories from fat. This is a very high-fat diet at 60% calories from fat. What we'll see with our diet, it kind of overwhelmed the system. So Celeste will tell you what she's been doing. We've modified this a little bit. But we split them into dietary treatments here at the baseline, and then either we performed a sham surgery or an OVEX surgery. And then every 4 weeks, we collected fecal samples. We used echo MRI to measure body composition. And then after 12 weeks, 24 weeks of age, we sacrificed the animals for tissues and blood samples. So not surprisingly, ovarectomy and high-fat diet was kind of a double whammy here. Increased energy intake, increased body weight gain pretty rapidly. So here's the energy intake at the bottom. We've seen this in dogs and cats as well. You remove the ovaries, and within a few weeks, you'll see differences in food intake if they're allowed to overeat. And so you can see this changed quite a bit, a pretty high percentage, and it maintained for a very long time at that level. The animals that were sham operated but had the high fat, they did end up getting up to the same amount of intake, but it took them about 8 weeks to get there. So there's certainly a trigger that's clicked here with the OVEX procedure. Not surprisingly, given the diets that were fed, that group, the OVEX high-fat fed group, split off pretty rapidly and were very different at 24 weeks of age. Of course, the sham operated high fat separated out as well, and then there's kind of a moderate change here on low fat. That the phenotype, it's with the low fat diet with only 10% fat, it's harder to develop that phenotype certainly. But we'll show you there are certainly some differences even in these animals here if you look at tissue samples and gene expression. So certainly, to go along with that, not surprisingly, fat mass was increased. This is echo MRI data at week 4, 8, and 12, and you can see certainly it increases pretty rapidly. And so this is percentage, so again, it's even more dramatic, but certainly the fat mass was increased quite a bit. Along with the overall fat mass, the adipocyte size increased as well. And so certainly in high-fat diets, it increased, but even there was an OVEX effect. Even in the low-fat fed, in the high-fat fed, there was an OVEX effect that occurred there. That translated also in this ectopic fat. If you look at liver triglycerides, certainly the one that sticks out is the OVEX group with high-fat fed. But you can see even here on the image, and we're not quite statistically there, but there's changes occurring even with the low-fat fed mice at this level too. So certainly the phenotypic changes that we would expect. Along with that, we did a lot of, I say we again, Zowin did a lot of gene expression analysis, and what we use is the Fluidigm Axis array. If you're not familiar with this, there are 2 different arrays. This is a 48 by 48 array. What this allows you to do, and it's targeted gene expression, so we develop primers for up to 48 genes, and so you can put the primers on one side, you put the samples on the other side, and in the middle, you have about 2,300 different wells, and this is all through microfluidics. You have 2,300 reactions occurring on this as axis array. So what you get from one axis array is basically a heat map like this. And so these are all the genes. Of course, you can't read all of those on the side, and then you have the treatments on the top here. It's not perfect, but certainly just shows you there's still some biological variation. But really, what you have is the OVEX high-fat group, the OVEX, the sham high-fed group, and then the groups here that were fed the low-fat diet. That overall, all of the gene expression that's there, you can see these patterns that they split off. And of course, in the paper, when we publish this, we will break it off into individual box plots. And so I've shown a couple of things here: adiponectin, leptin, TNF-alpha, some of the chemokine ligands that are related to inflammation. And so certainly, not surprisingly, lipid metabolism was altered in the gonadal adipose tissue, altered insulin signaling, glucose metabolism, inflammation. A lot of these things have been changed. And so we have really 4 tissues we've done this with. So this is gonadal adipose. We also measured subcutaneous adipose, liver, and as well as cecal tissue. So what Zowin did was really for adipose, we picked different categories of genes that are listed here, whether they are metabolically related or important, or whether it's inflammation or some other type of signaling pathways, and we put those on the Fluidigm for adipose tissue. The liver panel has some of the same genes, but then we have different genes for the liver, and then cecum, of course, is quite different. We have some inflammation genes, but there's a lot with tight junctions and other functions we think are important for that tissue set. And so it's a very gives you a lot of data, but it's a little bit more, it's targeted. So the good thing is you can pick the genes you know have an important function. They're all annotated. So it's a little bit different than RNA-seq or microarrays, where you might get signals back and differences, but you don't really know what the function of those genes are. So it's a way to kind of target that. Of course, we measured the gut microbiota. The main thing, again, we can see both ovarectomy and the high-fat diet, or the fat level of the diet, certainly altered cecal microbiota. And this is in this middle panel here. And of course, every dot, I think a lot of people know what PICO-A plots are now, but you look at the microbiota profile of these animals, and they each have a dot. And this is at week 24 of age. This is after 12 weeks of OVEX. What you can see here is the high-fat diet really split itself off from the low-fat diet. And so the blue dots here, and this is the unweighted, and this is the weighted at the bottom. The blue dots are the OVEX high-fat. The green dots are the sham high-fat. So you can really take a vertical line and split based on fat level. Then we have the red dots that are the OVEX low-fat, and then the sham low fat. And you can see in the unweighted, this is just, again, for those that don't know, the unweighted is the presence or absence of bacteria. So you can often see these shifts with some rare or low abundance taxa that will split them apart. If you look at the weighted, now you're looking at the abundance of the bacteria that are there as well. And so what you can see between unweighted and weighted, the high fat, this 60% calories from fat really, I think, overpowered the system. So you don't really see a difference here. But those eating low fat, you almost have a complete separation between the OVEX and sham group. And so it's an interesting shift there. What you'll also see is if you look to the right here, this is only at the phyla level, just due to time. I'm not going to go through the genera, but we saw certainly an increase in firmicutes versus Bacteroidetes. And so this firmicutes Bacteroidetes ratio increased, which has been shown. I question how valuable that ratio is, but people ask you all the time. It's been shown with obesity and high fat diets. And so we demonstrated that here in this study. More interesting to me, though, is this low fat OVEX group, that we have kind of a bloom here of the Actinobacteria, and its Bifidobacterium is the largest genera kind of driving this change, as well as the Verrucomicrobia. And this is where this is kind of claim to fame so far is the Akkermansia. That really is very much of a specialist in looking at mucosal, mucus degradation at the mucosal level. And so there's an interesting change there with OVEX and low-fat diet. From a diversity perspective, again, species richness was different between low and high fat with a reduction. It looks pretty tight here, but statistically it's different. The OVEX really didn't show a difference. And then with high fat diet, there was an increase with beta-glucuronidase. And this is, I have to give credit here, Matthew Redenbaugh from UNC Chapel Hill conducted this analysis for us and will be a co-author on the paper, certainly. But what we were expecting here, high fat diet will often increase beta-glucuronidase activity. And beta-glucuronidase activity in the gut is usually inversely related with estrogen concentrations. And so we thought that the OVEX group would have an increase in the beta-glucuronidase activity. It looked like it was trending in that way on the low-fat diet, but again, with the high-fat diet, I think overwhelmed that. So we didn't really see what we expected. But again, this is at the end of the study. And so I think what we really need to do is look earlier in that post OVEX response and some of the changes that are occurring earlier on. But certainly we saw some changes here. I know I'm going to run out of time, so I'm not going to give you much more data, but I did throw a few pictures here that of course you're not going to be able to read, but it just makes the point. We have all the other expression data for our other tissues. We have longitudinal fecal microbiota data that are somewhat interesting, and even at 4 weeks of age, they're already, or 4 weeks, sorry, after OVEX, they're splitting apart. And so I think some early intervention and sampling needs to be done there. And then, of course, we're looking at these different data sets and trying to look at associations. It will not be causal necessarily, but certainly will identify, hopefully, areas to target in future studies, looking at the phenotypic differences in these tissues, but the mRNA expression as well as the microbiota populations, and kind of comparing those with these to try to identify what bacteria might be driving some of these changes, and vice versa. Maybe some of these physiological changes in the animals are resulting in changes in the microbiota. You have to look at it both ways. Lastly, the last part of this project was really the inoculation of germ-free animals when Zhu Wen was in the laboratory at Wisconsin-Madison. At the time, we did not have a germ-free facility. We do now, but we didn't at the time. So we have some interesting data looking at the donors that were OVEX or sham, and so in these two groups, and how they led to changes in the germ-free recipients. And so I'm not going to show any data there. It's unpublished, but certainly you can be watching for that, and hopefully we can kind of wrap this up here and be publishing it soon. Before I just want to acknowledge a couple people, I'll certainly just say that I think there's some future needs here. I work with ILSI North America on a few projects and advisory committees. I think what we need to do is, from a human perspective, we need more and more longitudinal study designs and repeated measures. I think we can learn some with spot sampling, but we really need more work in that area. Controlled and monitored dietary intake. A lot of people are working that, and ILSI North America has some committees that are working on that with some experts in the field. I threw up this image here of the NIST workshop, and so the National Institute of Standards and Technology, Scott Jackson and his group there are working on standards for microbiomics and metabolomics. And so this was about a year ago. but there's some work being done here. I think one of the challenges in this area is every population is unique, and so making comparisons across studies very difficult without having standards. And I think if we get some standards in place, I think that'll help out quite a bit. Similarly, in the animal models, I think we need to understand more and more at the microbial gene level. How is it relating to phenotype and what's driving phenotype, and going back and forth. Again, controlled environment and feeding, again, stressing the need for semi-purified diets, or at least avoiding phytoestrogens that are naturally in food components. Or if you want to study those, do that, but certainly if you're not trying to study those, you have to have them out of there because they're going to be biasing your results. And then I think there's a lot of germ-free models that can help. Lastly, I think just as a lot of the bioinformatics folks are getting on board and collaborating with nutritionists, I think we have to involve more and more of the microbiologists and looking at the genes, the gene level, metabolite level, and try to understand what is being transformed, what's being recycled, and how is this being done, and then how is it affecting host phenotype. With that, again, I want to thank a few people. I have a lot of names on here, but my current laboratory, I have a lot of great people that are working with us, a lot of nice collaborators and very great collaborators at the U of I. I didn't really touch about the pet microbiome, but I've had a lot of sponsorship there. And then on the human and rodent side, some internal programs, certainly at the U of I, whether it's Division of Nutritional Sciences or the College of ACES, getting some seed funding that has led to other external funding. I do have a professorship as well, and so while Kraft Heinz isn't providing a direct research for that, there is some funding that comes with the professorship that I've really pushed toward this area. And so again, General Mills, Ingredion, and PepsiCo have been working in the past on the human side. And so with that, hopefully I'm not too much over time. I get too excited and try to add too much data. But with that, I'll turn it over to Celeste. And then at the end, I'll be happy to answer any questions that people have. So thank you for attending.
Great, thanks so much, Dr. Swanson. And as mentioned, we will turn things over to Celeste Alexander. And by way of introduction, Celeste is a fifth-year PhD student in the Division of Nutritional Sciences at the University of Illinois, and she is, of course, under the advisement of Dr. Swanson. We also wanted to give a big congratulations to Celeste, who just recently defended, and today she'll be talking to us about some of her thesis work. So with that, we'll turn it over to you, Celeste.
As per usual, I was muted when I started talking. I swear, no matter how many months we're in quarantine, I will always start by doing that. But thank you for the lovely introduction. And I'm really excited for the opportunity today to talk to you guys about some of my dissertation work. As Kelly mentioned, I am kind of working on an offshoot of the OVEX model that has been previously researched in our lab, focusing on cholecystectomy and its effects on metabolic and gastrointestinal health. So I'll be talking to you a little bit about that today. Just as an overview of what I'll be chatting about, I'll provide a little bit of background about cholecystectomy, as well as bile acid signaling and microbial metabolism of bile acids, briefly introduce the novel dual surgery model that we developed at the beginning of my PhD, and then mostly talk about my main dissertation project that I've split apart into kind of the impacts on metabolic health, as well as GI physiology in the microbes and a potential role of the microbes on these host outcomes. So cholecystectomy, or XGB, as I'll refer to it most of the presentation, is the surgical removal of the gallbladder, which is this organ. And this is most commonly done in response to chronic gallstone formation or gallstone cancer or gallbladder cancer. And it's actually the most common abdominal surgery that's performed in the United States, with over 800,000 being done every year. So there's millions of people in the United States living without a gallbladder. And these people generally tend to be women who are white, Caucasian, middle-aged, and overweight or obese. And I've highlighted the female and middle-aged part of this demographics, as this relates back to the menopause model that I'll get back to in a couple of slides. So normally, the gallbladder functions to store and concentrate bile that's produced by the liver until the consumption of a meal that contains fat. Once we eat food that has fat in it, the gallbladder contracts, bile is released into the duodenum, where it aids in the emulsification and absorption of dietary fats, fat-soluble vitamins, cholesterol, things like that. However, once the gallbladder is removed, bile is then continuously released directly from the liver into the duodenum as it's made. And so what happens is we have chronic exposure of the gut to smaller volumes of less concentrated bile, as opposed to a few times a day large boluses of concentrated bile. And so this exposure of bile in the duodenum continuously can result in something called bile acid-induced diarrhea. And this is a common symptom associated with XGB patients. However, other than this symptom, XGB has long been thought for decades to basically have no long-term complications following this surgery. However, recent research is suggesting that potentially the opposite is true, with clinical and epidemiological studies suggesting an association between XGB and various metabolic and gastrointestinal diseases like cardiovascular disease, metabolic syndrome, non-alcoholic fatty liver, colon cancer, etc. And so it's hypothesized that the reason why these phenotypes may be emerging following cholecystectomy is the constant recycling of bile acids from the liver to the gut and back, and exposure of the gut chronically, particularly during fasting states where normally bile acids would not be present in the gut. And so they call this bile acid flux, which has been shown both in humans and in animal models for decades to increase following cholecystectomy. So the number of times the bile is being recycled through the body per day greatly increases. And so the reason we think this flux may be driving these phenotypes is that bile acids are very potent signaling molecules, or arguably hormones. And they bind a number of cell surface and nuclear receptors that are expressed throughout the body, namely FXR and TGR5 are two of the biggest ones. And these bile acids can bind them not only with different affinities, depending on the bile acid species, but also with different effects. You can see here that some bile acids may act as agonists of FXR, while some may act as antagonists. And so differences in the bile acid pool composition may result in different signaling pathway activation. And so just to give you an idea of the types of pathways that are regulated through these receptors, I've just put them here. They're very potent inhibitors of their own production through a feedback inhibition mechanism, but they are capable of regulating processes related to energy metabolism and immune response. In addition to acting as hormones, bile acids may also serve as substrates for microbial metabolism, as well as acting as antimicrobial relationships. So it's kind of a unique relationship between bile acids and microbes. So while some microbes are sensitive to bile stress and may be killed off by the presence of bile, other microbes possess enzymes that are capable of metabolizing these bile acids to make them less toxic to themselves. So I'll just give a little overview of bile acid metabolism to give you an idea of just how much diversity in the bile acid pool can result from the gut microbiome. So if we start out from what the body produces, we have bile salts, and these are bile acids that are simply conjugated to glycine or taurine, as Kelly alluded to earlier. And once they're released into the duodenum, a number of major species within the mammalian gut microbiome possess bile salt hydrolase enzymes that can cleave off that glycine or taurine, leaving us back with just our primary bile acids. Once the glycine or taurine is removed, primary bile acids can be acted on by a multi-step enzymatic pathway called 7-alpha dehydroxylation that produces secondary bile acids. And these are considered to be more toxic to the host because they're more hydrophobic. And despite the fact that a very small portion of the gut microbiome is capable of producing the enzymes to do this reaction, secondary bile acids still remain the primary bile acid species found in stool samples in mammals, indicating this is a very efficient process within the gut microbiome. And then finally, both primary and secondary bile acids can be acted on by a host of hydroxysteroid dehydrogenases that produce different iso, oxo, and epi bile acids that we aren't going to get into here. But just to give you an idea of the diversity of bile acids that can be created through microbial metabolism. So taken together, we know that bile acids are potent signaling molecules that can be extensively metabolized by the gut bacteria. So one could hypothesize that a perturbed microbiome may result in compositional changes in the bile acid pool, thus, altering the metabolic signaling pathways through that change in bile acid pool. So in addition to XGB resulting in an increase in bile acid flux, we hypothesize that potentially a perturbation to the microbiome and bile acid pool may be also driving some of these phenotypic changes. So I mentioned on the first slide that the majority of people that are getting their gallbladder removed are women who are around the middle age. And this is important because the median age of XGB corresponds to the average onset of menopause in women. And as Kelly has talked about for a while, so I don't have to go into many details, we know that menopause results in a lot of body compositional changes that result in increased metabolic dysfunction, obesity, et cetera. And we can see that, in a postmenopausal age frames, the prevalence of metabolic syndrome goes up in women. So women that are getting their gallbladder removed and then progressing through menopause may have a double hit to their metabolic health that has yet to be investigated. Again, as Kelly alluded to, so many preclinical research studies are done specifically in male mice, and this is no different. There have only been a few preclinical studies done in XGB research in general, and recently all of them have been done in male mice, despite the fact that over 70% of XGBs are performed in women in the clinical population. So we didn't want to just study XGB in female animal models. As we know, sex hormones are very important, and we know that the majority of women undergoing this surgery are going into menopause. So we decided to also utilize the ovarectomy model as part of the research in order to kind of develop a clinically representative animal model to better study this surgery. So in order to do this, no one had done it yet, and so we developed a novel model, and I say novel, it's a novel dual surgery model that hasn't been investigated, where we've done both the XGB and the ovarectomy procedures. And so the reason we wanted to do this is to, first and foremost, determine the feasibility of doing a dual surgery. And by feasibility, I mean, do the mice live? That's kind of an important part of the research. And so we XGB'd our mice all at baseline, and then half of them either went under ovarectomy or the sham procedure where they keep their ovaries. And so we did have good survivability, over 90% in both of our surgeries, so this was good. And then secondly, we were wondering and wanted to determine whether or not OVEX presents with the same phenotype in an animal that doesn't have its gallbladder as we know it does in an animal that does have a gallbladder. And so we wanted to see that OVEX behaved the same way, and we have plenty of research in the literature and from our own lab to go off of. So the long story short is kind of, in some ways we do see that with, for example, with regard to body weight gain. You see up on top, the ovarectomized animals gained significantly more body weight over the course of this pilot study compared to those that were sham, although the increase in body weight gain appears to plateau between the groups after about 14 weeks. But when we look at other aspects, such as targeted gene expression throughout the gastrointestinal tract, we don't see similar gene expression profiles as previous data from our lab in ovarectomized versus sham mice. And we also don't see similar patterns in the fecal microbiota composition. And I've just put up here the most abundant genera. So this was leading us to believe that XGB might be having some kind of masking effect or interactive effect with the ovarectomy, particularly related to gut outcomes, that was preventing us from seeing that kind of traditional OVEX phenotype. And so after we developed the surgery model, we wanted to then identify the independent effects of XGB on a variety of different outcomes. Additionally, as a nutrition lab and one that studies fiber, we were interested to see if we could also incorporate some kind of dietary intervention as a therapeutic strategy. And we were initially drawn to psyllium husk, and this is because it's a soluble, lowly fermentable fiber with very strong bile acid binding capacity. We know it's safe in the clinical setting as it's widely used for a variety of different situations. And we also know in preclinical studies that psyllium husk may have protective effects on the gastrointestinal tract. When incorporated into the diet, psyllium husk can protect against chemical-induced colitis, so may have added benefits there. However, we didn't want to just include psyllium husk into our diet. We were, as our driving hypothesis for why XGB may be leading to these associated disease phenotypes, is our hypothesis is that it's the bile acid presence in the body and through flux during fasted states is what's causing these diseases to arise. We wanted to specifically test if postprandial consumption of psyllium husk may be able to attenuate the effects of XGB. Essentially, our hypothesis was if during fasted periods, if we give psyllium husk, can it sequester those free bile acids in the gut and prevent them from having aberrant signaling going on during fasting stages. And so this was a second component of our research in my main dissertation project that we incorporated. So the overall objective of my main research project was to determine the systemic impact of XGB and the postprandial psyllium consumption on metabolic health, GI physiology, and the microbiome using our model that we developed. And we hypothesized that XGB mice would exhibit a disease phenotype, a metabolic disease phenotype, as well as showing altered gut histology and microbiome compositions and an increase in secondary fecal bile acids. And that, overarching, that the psyllium husk would be able to attenuate, at least in part, some of these outcomes. So for our study design, we utilized a 2x2 factorial design in which we had 2 independent variables. The first being, surgery, so either the XGB surgery versus the sham surgery. And in this case, the sham surgery refers to, leaving the gallbladder intact. And then our other variable was whether or not animals were on the psyllium husk intervention versus the fasted intervention, which I'll talk about on the next slide. We utilized the same type of mice, had a great survival rate again, which was promising, and we put our mice on a high-fat diet. As Kelly mentioned, we utilized the 45% high-fat diet because we want to more closely mimic, kind of a westernized type of diet, but 60% as typically used in rodent studies is a little bit extreme. And so we wanted to use one that was a little bit more representative of, a high-fat diet in humans. So our timeline, at baseline, after an adaptation period, we randomized our mice to undergo either the XGB procedure or the sham XGB procedure. So they underwent the same surgical stress, just kept their gallbladder. And then at the same time, further randomized the mice within their surgical groups to either be on the psyllium husk intervention or the fasting intervention. 4 weeks later, all 4 groups of animals were ovarectomized in order to mimic that progression into menopause. And then the study was carried out through 16 weeks, at which point the mice were sacrificed for tissue collection. And then throughout the course of the study, we collected fecal samples for microbiome and bile acid analysis. We perform echo MRIs for body composition, and then we also collect food intake and body weight data as well. So I mentioned we had a fasted group. And so in this case, to implement our dietary interventions, we employed a modest time-restricted feeding schedule, and this was twofold. One, the obvious, if you want to have a postprandial diet intervention, you have to have a time where food is not available. So traditionally in rodent studies, ad libitum 24-hour-a-day access to food is pretty typical. So we needed to make sure we had a period of time where food was not accessible to these mice. The second reason is we think that this pattern and this feeding schedule that we used would more closely model human eating patterns. While mice, eat more in their diet cycle, but really eat kind of the whole day, humans tend to eat more, structured meals that are a little bit spaced out. And then there's a longer period of time when we're sleeping, for example, where we're really not consuming anything. And if we're studying XGB research, I argue this is a large strength of our study because the driving hypothesis, as I've mentioned, is, the bile acids present in the gut during a fasting state, which in a traditional rodent feeding, method is maybe not achievable. So we utilize this kind of feeding schedule. So it looks like a day in the life of our mice. And between the 2 groups, the psyllium or fasted, they were identical between 5 p.m. and 8 a.m. the following morning. They had ad libitum access to the exact same, high-fat diet that I mentioned on the previous slide. But at 8 a.m. for the fasted animals, food was removed and measured, and then they simply just had access to water until 5 p.m. when food was then reintroduced. And in the psyllium group, food was removed at 8 a.m. and instead was replaced with these psyllium husk pellets that we formulated for the study, and they had ad libitum access to that. At 5 p.m., the psyllium husk was removed, weighed, and then food was reintroduced. So as I mentioned, we measured food and psyllium intake daily for the course of the study. So to kind of break the study results down into sections, I'll first highlight a few of the metabolic health outcomes we looked at before moving into more of the gut side. And the first thing that we look at is body weight and food intake. And we found that our XGB mice, both groups over time, gained significantly more weight compared to our sham groups. And this was despite the fact that they were eating relatively similar amounts of food each day. We can see here the XGB fasted group did consume more food over the course of study than the others, but the XGB psyllium group is consuming almost identical amounts of food compared to the sham animals, despite gaining significantly more weight over the course of the study. We saw that this body weight increase was due to an increase in adiposity. If we look at body composition change over time, we see that by week 12 and 16, the XGB mice had gained a greater proportion of body fat mass compared to our sham animals. And if we look at individual adipose depots, we look at 4 major ones: the subcutaneous, gonadal, mesenteric, and perirenal. We see that this increase in adiposity is fairly well distributed around the body in these major adipose sites. There isn't, they were all significantly greater in the XGB mice. Additionally, we see ectopic fat deposition with the XGB mice. having a significantly greater liver triglyceride content compared to sham. And this is concurrent with a couple clinical trials that are coming out showing increased hepatic steatosis in people following XGB. We also looked at blood lipids and total bile acids at fasting, and we see that XGB increased fasting plasma total and LDL cholesterol concentrations, as well as fasting plasma triglycerides, with just a tendency to increase HDL. And we did see that psyllium husk had an independent effect on both LDL and triglycerides, lowering those concentrations independent of surgery. Surprisingly, we did not see any differences in total fasting plasma bile acid concentrations, though we do see quite a bit of variation, particularly in our fasted groups. So just to briefly summarize that, again, our control group here are the sham animals, which are just a postmenopausal animal with an intact gallbladder. And we're seeing that XGB on top of that is exacerbating postmenopausal metabolic dysfunction. We see that in increased adiposity and ectopic fat deposition and an increase in plasma lipids. And psyllium husk intervention here only attenuated blood lipid levels, a few things. And I put in here an asterisk at 8% of daily intake. This is by weight on average, and I do think this would be an overestimation, as if you saw the picture, the psyllium husk pellets were pretty crumbly. So recovering all of the unconsumed psyllium husk was a challenge sometimes if they really crumbled it. So it may be a little bit of an overestimation of actual intake. So if I move into more of the gut side, the microbiome, which is why everyone's here, again, we hypothesized we would see an altered gut histology and microbiome composition and an increase in fecal secondary bile acids. We did not see any differences in gut morphology. We looked at crypt depth and villus height and did not see any differences throughout the gastrointestinal tract. But we did see some differences with respect to mitotic rate in the gut. In the cecum, XGB significantly increased mitotic rate compared to sham. And in the ileum, the psyllium husk had an independent effect where it increased mitotic rate as well. So mitotic rate can be kind of a double-edged sword with respect to gut health. On the one hand, aberrant mitosis can be indicative of a precancerous state, whereas, if we're talking about the gut, especially the epithelial layer, constant sloughing and regeneration of that epithelial lining is associated with, gut health. So further research would need to be done to identify the physiological relevance of an increase in mitotic rate in this particular circumstance. We also looked at gut immune cell populations throughout the GI tract, and we found a number of different changes, but one of the things we were most interested in was a diminished proportion of CD8 positive gamma delta T cells. And this is a unique subset of T cells that of gamma delta T cells that has recently been characterized. And what was interesting to us is it has been found to be diminished in biopsies of patients who have inflammatory bowel disease as well. We also stained our histology sections for goblet cells, and these are the mucus-producing cells within the gut, and bile acids are known to stimulate mucus secretion. So we hypothesized that a long-term adaptation of the gut to this kind of chronic bile acid stress following XGB might be to increase, the number of differentiated goblet cells within the GI tract. And so we stained just to look at the number of mature goblet cells. And we found that in the duodenal villi, there tended to be an increase in goblet cell density following XGB. But in the crypts, we saw a surgery by diet interaction where our XGB fasted mice had the greatest goblet cell density compared to the other three groups. But we did not note any differences in goblet cells in the other regions of the GI tract. Of course, we looked at fecal bile acids, and we did this longitudinally. I'm just, for time's sake, giving you a snapshot here. We looked at about 11 different bile acid species, and we did this longitudinally. So we looked at week 2, week 4, and week 16. And what we noticed short term was that the XGB mice had, independent of diet, had a significantly greater proportion of secondary bile acids compared to their sham counterparts. And the reason we looked at proportion was because there was no differences in total bile acids at any of the time points between the groups. And we see that this increase in secondary bile acids is largely due to an increase both in isolithicolic acid and lithicolic acid in our XGB mice. And what's interesting, when we look at week 16, so our long-term measurement, we look at secondary bile acids, we see that our XGB fasted group now, there's an interaction, and this fasted group in XGB has the greatest proportion of secondaries, with the other three groups looking more similar. But what's interesting, so we're maintaining this elevated secondary bile acid proportion in this group. However, it looks like the composition of the secondary bile acid pool is changing. So we no longer see at week 16 any impact of XGB on lithocholic acid. And in fact, we see a decrease in lithocholic acid in our XGB mice. So interesting that there might be a shift in kind of the composition of just the secondary bile acid pool. So getting more into the microbiome now, one of the first things we wanted to look at was whether or not XGB resulted in small intestinal bacterial overgrowth. There's been a few clinical trials that utilize breath hydrogen tests and indicate that those who have undergone XGB have an increased diagnosis of small intestinal overgrowth. And so our hypothesis for why this might be happening, as I've mentioned a couple times, in a normal state, we have large volumes of concentrated bile entering the gut a couple times a day when we eat. And because of the antimicrobial properties of bile, this may be enough to kind of keep the bacterial load in the small intestine in check. So a few times a day, there's just kind of this like kill off, of some bacteria in the small intestine as this bile comes through. However, in the case of XGB, we now have smaller volumes of less concentrated bile that may not be sufficient enough to keep that growth at bay, resulting in overgrowth of bacteria that would otherwise have died off. And so in order to test this, we cultured small intestinal contents from the duodenum and the ileum, and we cultured both aerobically and anaerobically just to get an idea of total CFUs per gram of content, just to get a rough estimate of the biomass in each of these regions of the gut. And so the first thing that we noticed, much to our surprise, is that XGB might not be resulting in overgrowth. In fact, in the duodenum, our total anaerobic counts tended to be lower in our XGB mice. And we did not see any effect of surgery on total bacterial growth in any of the ileal measurements, although we did see a tendency for psyllium to decrease anaerobic growth. So this led us to wonder, maybe this increase in SIBO diagnosis is not due to, increased biomass within the small intestine, but rather a robust change to the composition or functionality of the small intestinal microbiome. And we did sequence the microbiome. We are unfortunately kicking ourselves that we did not save contents to sequence the small intestinal microbiome, but we did look at the cecal and the fecal microbiome in these mice. And so when we look at the 16S data, we first look at measures of alpha and beta diversity. And across multiple metrics of alpha diversity in these mice at the end of the study, we see that XGB is actually resulting in increased alpha diversity, which is interesting. There was a recent study in humans that came out retrospectively, found that people who had undergone XGB had lower fecal alpha diversity compared to those who had not undergone XGB, so it was a little contradictory. And then with our beta diversity, Kelly already kind of explained how to orient yourself to these plots, so I won't go back over that, but we're obviously looking for clustering. We did not see any clustering by surgery here, but what we did notice is that our psyllium husk groups tended to separate. So we see here, with the respect of this one sample here, we kind of have, two separate clusters of our psyllium husk animals, and we have not quite identified what might be pulling those two apart. But in the weighted unifrac, we did not see any clustering whatsoever. When we look at these metrics for the fecal samples, we no longer see an effect of XGB on alpha diversity with any of our metrics. And this may indicate that, the XGB surgery is having a more robust impact on the upper region of the GI tract compared to the colon. And in these beta diversity plots, the enlarged circles are the ones that are indicative of the end of study, because we did collect these samples longitudinally. So it's a little difficult to see, but we don't have any clustering in either unweighted or weighted between our four groups at the end of the study. We were intrigued by this kind of cloud of psyllium husk animals in the unweighted unifrac that we hypothesized might be due to, just at the time that those samples were collected, maybe those mice just happened to really go to town on those psyllium husk pellets. But when we stratified the mice by high or low psyllium husk consumption and then colored the samples by that stratification, it did not separate out the way that we see here. So we're still trying to understand what might be pulling these samples out from the rest of the group. But by group, we did not see any distinct clustering at the end of the study. So after that, we looked at just the taxa level to see if there was any differentially abundant genera in both the cecal and fecal samples. And in the cecum, we saw a number of different bacteria, 12, that were differentially abundant either by, due to surgery or diet or both. But we do see a number of changes due to the XGB surgery here in the cecum. But I'll draw your attention to two in particular, which was the relative abundance of Lactococcus and Rumina Clostridium 5 that were more robustly impacted or increased in the XGB mice compared to sham. And I'll touch back on them, these two bugs, in a couple slides. When we look at the fecal samples, however, we saw a lot fewer differences, and only 2 of these genera were impacted by the XGB surgery. Most were a result of an impact of the psyllium husk. And so this kind of goes along with this idea that we've been starting to get of, you know, maybe the surgery is really impacting the upper GI tract a lot more than it's impacting the lower GI. Which kind of makes sense. You know, this is where the bile acids are being released. They're still pretty effectively reabsorbed in the small intestine. So maybe this is more of a local impact in the small intestine rather than throughout the whole GI tract. So to summarize the GI physiology and microbiome, we saw that XGB's increasing cecal mitotic rate and decreasing proportion of CD8 positive gamma delta T cells in the colon, which have also been reported to be diminished in patients with IBD. We also saw an increase in fecal secondary bile acids as we hypothesized, but what's interesting is it doesn't appear that the composition of the secondary bile acid pool is maintained long term. It does appear to shift differently than it was in the short term. We do see that XGB is influencing the GI microbiome. Contrary to clinical trials and our hypothesis, we actually saw decreased total anaerobic growth in the duodenum following the surgery. And we do see an increase in alpha diversity due to XGB in the cecal samples, but not fecal. And so, you know, as I've mentioned, this kind of trend, we're noticing that there may be a larger impact on the upper regions of the GI tract. Unfortunately, our psyllium husk intervention was insufficient to ameliorate any of the effects of XGB really in this particular set of outcomes. But unsurprisingly, this is dietary fiber. It had a lot of its own independent effects. So like I said, this is very unsurprising. We know dietary fibers have a robust impact on the microbiome and gut health. So this was not a shocking finding to us. So one of our kind of secondary outcomes after we looked at all of these different things was, could we just run a basic correlative analysis and identify potential microbe-dependent mechanisms that might be contributing to the observed phenotype. So what we did is we took all of the bacterial genera that were differentially abundant in both the cecum and fecal samples, and then we correlated it with host outcomes that were also differentially abundant to see if there may be a relationship there. And we hypothesized that outcomes like body weight, immune cell populations in the gut, and fecal bile acids would be more strongly correlated with the microbes than the other outcomes. And so for some of our initial observations, to no surprise at this point for us, the outcomes that were much more strongly associated with the cecal microbiota than the fecal microbiota, again, indicating more of a robust impact upper GI. And much to our surprise, the bile acids were not strongly correlated with differentially abundant cecal or fecal microbes. If you see here, there are very few just weak correlations. And so that was a bit of a surprise, but may allude to the fact that, you know, for especially looking at secondary bile acids, you know, we're not going to capture that at the genus level because the bugs that are capable of, you know, doing secondary bile acid production are in such low relative abundance, they would be not captured by our sequencing data. However, when we looked at cecal microbiota and our metabolic outcomes, a few bugs stood out to us immediately. And this was Lactococcus and Rumina Clostridium 5. As I mentioned before, you know, those two were dramatically increased in our XGB mice compared to sham. And we see that they're moderately to strongly correlated with a number of outcomes related to metabolic dysfunctions. We have body weight, you know, fat mass, adipose mass, blood lipids, and liver triglycerides as well, indicating that maybe these bugs are playing a role in the development of that metabolic syndrome or metabolic disease phenotype. When we looked at the cecal microbiota and gut physiology outcomes, a few things stood out to us as well, namely that Bacteroides was negatively correlated with cecal mitotic rate, and Lachnoclostridium was correlated with, differentially correlated with cecal and colonic T cell proportions, indicating that maybe this genus is interacting with the host immune system. So just as a brief summary, again, we did not see any correlations with the microbes and the bile acids, much to our surprise. However, we did see some stronger correlations between a couple of bacteria in particular in the cecum and metabolic dysfunction, as they were positively correlated with a number of these outcomes. And this is interesting to us because identification of these microbial taxa that might be contributing to that metabolic disease phenotype might aid in microbiota targeted therapeutic strategies unique to this population, for example. And it will also help us to shape future notobiotic studies that will help us to determine a more causal role between changes in the microbiome and the outcomes that we're seeing following XGB. So just to kind of summarize everything that I've talked about, first and foremost, I know I just talked about it for one slide, but a big part of this work was developing a clinically representative model of XGB. And to our knowledge, this was the first study to explicitly look at XGB in female animals and the effects here, not to mention a female menopause model. And this is concerning, as I've mentioned, because over 70% of these procedures are done in women. And as Kelly's alluded to, these sex hormones play a major role in metabolic health outcomes and in the microbiome. Also, to our knowledge, we were the first study to investigate the impact of XGB on GI physiology in the microbiome in detail. There's been some studies in animals and mice looking at fecal bile acids or transit time or colon cancer incidence, but not looking at, immune cell infiltration, histology, and things like that, and gut microbiome. And then kind of last, while this is only one study, our data, along with emerging research, both in clinical and epidemiological trials, are suggesting or challenging this notion that XGB has no long-term complications. So just given the vast number of procedures that are being done every year and how little information we know about the impact of this surgery on human health just highlights the need that we need to continue understanding exactly what's going on following the surgery long-term through use of clinical and preclinical trials. In the future, we would love to identify mechanisms that are driving this phenotype, and this is something that I'm currently working on. Unfortunately, due to COVID, it was really delayed, so I don't have the data yet. But we're doing, as Kelly mentioned, we did a fluidigm access array, so we have a huge panel of targeted genes in the duodenum, ileum, and liver that we're looking at to try and develop kind of a proposed mechanism for what might be going on based on that data. We also, as the point of this talk, would find it really interesting to investigate the sex differences in response to XGB. Do a postmenopausal female mice behave similarly in response to XGB as conventional male mice? But running a study with all three groups would be really interesting. As nutrition scientists, obviously, we are interested to see if the exploration of or looking at different dietary compositions can maybe mitigate the development of the XGB phenotype. And then last, because I've said this 50 times, but I'm so mad we didn't do it, to identify the changes in the small intestinal microbiome. I think our data really suggests that there is, a more robust impact here up in the small intestine that could be really interesting to take a look at. And so with that, I just want to thank, obviously, Kelly, my advisor, for supporting me to do all these surgeries and add another one into the mix, and our labs and collaborators and funding sources, and obviously DNS, ASN, and ILSI for the opportunity to talk to you guys today. So thank you, and I think Kelly and I are going to take questions now.
Great, thanks so much, Celeste. Yeah. Okay, we do have a few questions. So I think we'll start just somewhat big picture, and we wanted to ask, you mentioned earlier, Dr. Swanson, that our ability, for the most part, we use fecal samples for humans, but we have the ability to look at different areas of the GI tract for animals. So the question is, how much more do we know about animals compared to humans with respect to the gut microbiome? Is it, we have 10 times more information, or are we basically really early stages in all respects?
I guess I would say we're pretty early stages on all respects. Again, hopefully in the future there will be procedures where we can. I know people are working on smart pills or some other way to get samples while it's in the large intestine, or if you're looking at disease, maybe in the small intestine. But I think we're somewhat hamstrung a little bit if we're relying on the fecal samples all the time. I think especially when you get to metabolites as well. So a lot of people have moved kind of through transcriptomics all the way to the metabolite. But a lot of metabolites it's the end result. The great majority, 95% 20% or so of the short chain fatty acids, for instance, are absorbed in the gut, and so you're left with the very the products that haven't been absorbed. A lot of them are volatile, so if they're not collected and stored properly, they're gone, and so you can't measure those. And so there's a lot of I think methodological issues that need to be tackled here, and that's where getting standards and standard procedures. I'm on several advisory groups, and if you say, how do you collect your samples, how do you store them, you oftentimes get a lot of different responses. And so again, it's difficult to compare across studies, but it's hard to put a value on it, I think. But HMP, the Human Microbiome Project, that was the gut sample was a fecal sample, and they were very, I would say, they were all healthy individuals. There've been, since that time, there have been a lot of studies being done, but again, relying just on fecal samples, we're going to always be limited if that's all we have to work with. I think it can be somewhat predictive, I think, of what's in the colon, but certainly anything up into the small intestine, it's not going to relate at all. And even in the colon, I think we have to, if we can understand activity, that's the key, and what those interactions are with the gut, what the interactions are with each other. It's a really difficult challenge, because once you get to that fecal sample, and again, in our studies too, if you're measuring just the DNA, the abundance of those, it has nothing, or very little probably to do with the actual activity of the bacteria and actually what they're doing. So it's I'm not sure if I can really answer the question, but there's a lot. It's still very, very early in the game. If anyone, I always say, if anyone says they have it all figured out, I'd walk away slowly, because no one really has this figured out. It's going to take a lot of time, but also the molecular microbiologist all the way up to the people doing the epi studies in humans, and everywhere in between, I think, is really to tackle some of these questions. And then on this topic here, we're talking about sex hormones, we're talking about the microbiota. There's so many interactions. It's pretty complicated.
Great, thanks, Kelly. Yeah, and I guess it's especially complicated to standardize the way things are done here, because this matrix that we use, the fecal sample, is unlike anything else. And you and I know NIST is working on developing the standard, but for them, it's a completely new type of matrix to deal with. So we'll get there eventually.
Yeah, exactly.
Yeah. So, okay. So this is a question about the hypothesis that you presented in the introduction, where women do this catch up with men at the point of menopause. So the question is, supposing, well, there's a lot of cultural pressure for women to stay thin, to maintain weight that maybe men don't have. So is there some other reason that you could think of why there's that catch-up point, that intersection after menopause?
Well, I hope my wife isn't watching, but I do. I get in trouble sometimes. Sometimes I deserve the abuse I get at home, but other times I don't deserve it at all. But an older gentleman, I'll just say, might be called handsome, but she says that's not how we treat women as they age. So it is a little bit different. And so culturally, whether you look at the movies and or just commercials on TV, whatever you see, or just in at home or in the community that you're in, there are differences there. And so and you're right that could certainly be a factor. It certainly is a factor, I would say, not maybe, but it certainly is. Where you see, though, certainly in animal models in particular, and we've shown it across, whether it's rodents or dogs and cats, there are differences, though, when you take estrogen away. And experimentally in an animal model, it's more abrupt, certainly, in what Celeste did. It's a very abrupt change, but certainly you see a lot of those changes. So I think certainly there could be some cultural things going on here, and pressures in society, I suppose. But I think it's pretty easy to prove as well that certainly hormone changes result in a lot of changes with metabolism. And again, as we study this more, I think you're going to see that then the microbiota are altered as well. I think a question, though, that I always think about, is it a direct, and I think about it from a diet perspective as well, is it a direct effect? Is it a substrate that they're not getting, or now that they do get and now they're modifying? Is it indirect? So estrogen changes the metabolism of the host, and now the host sends different signals to the microbiota. So there's a lot of things going on there that I even think, again, I think about it that from a dietary perspective as well. Some people say, well, we shouldn't eat a lot of dietary, sugar in our diets, and there's going to be a microbiota change. Very little, if any, of that sugar, if it's pure sugar, sucrose and monosaccharides, disaccharides that are digestible, are ever going to reach the hind gut anyway. But you might show a change in that microbiota population. It's probably not a direct effect. It's some other effect, most likely. And so we have the same interplay here. I think it's actually in some ways maybe it's more complicated with hormones than the hormones you can control them somewhat, but diet is much more controllable, and so it's tricky to study. But the point is well taken. Certainly the societal pressures certainly could contribute to that catch up that happens around menopause.
Right. Okay. We'll shift to a question about Celeste's work. So the question for you, Celeste, is what are the dietary recommendations for cholecystectomy in those patients? And have you studied people or mice that consume those recommended diets versus the high-fat diet that is Western?
Yeah, my understanding with my limited clinical experience is that, and also just from like talking with people, so this is totally anecdotal data that I have, but that the recommendation following XGB seems to be fairly inconsistent. I think it depends on, your doctor, and I think maybe their level of nutrition training. But obviously when we talk nutrition scientists or dietitians, that, the gold standard is kind of, if you have issues handling dietary fat, it's to either like lower your dietary fat intake or eat just smaller meals throughout the day. So the amount of fat you might be consuming is different. But a large number of people don't have any problems. It's hard to find any like data that people have collected on like, what have you had your gallbladder removed? What are you eating? How much, like, what are your dietary practices to see what kind, are people following these dietary patterns, or have they had to adapt their diet? There's been some research. Again, it hasn't been done in like decades because they did it like twice and then assumed everything was fine. But they actually did not find any statistical difference in dietary fat malabsorption between people with gallbladders and without. So statistically speaking, they don't have dietary fat malabsorption, but if you look at the variability in people who have no gallbladder, it's like dramatically higher than those that do. So I think that it could really be individual response, where some people may, for whatever reason, have issues handling fat afterward versus others. And theoretically that could involve the microbiome. So I think that's a really interesting question. But my understanding is that it's fairly inconsistent, the recommendations that are given to people.
Okay, yeah, maybe it's a point for personalized nutrition and individual experimentation. But would you say that dietary fibers or a probiotic or a prebiotic could generally be beneficial for gallbladder removal patients?
Yeah, I think at this point it's really hard to say, just because there's so little research that's been done on even the changes in the microbiome following this surgery. So there's like 3 papers ever in humans that have even measured the microbiome, and they've all been like case cohort. It's never been like follow people before and after, and so I think that it's hard to say because, in the theory, like there may be some kind of benefit, but there is so little research out there right now to even give an educated, recommendation on what might be beneficial. So I think before we're even able to get to answer that question, we need a lot of more large scale studies to understand what exactly might be going on in the microbiome with these patients, which in theory shouldn't be hard to gather that data since there's millions of people that don't have a gallbladder. You'd think it would be a pretty large population you could draw from. So, but I'm hoping, that research starts coming out. There's just like 3 papers right now.
Okay, wow. Okay, and so this question, I think it gets to Kelly's point about the complexity of all of these questions. So the question is, do the effects of hormonal changes, for example, menstruation, depend on the initial composition of the gut microbiome? So I think the question is about, there are all these different things that affect the gut microbiome: diet, rural versus urban living, mental health. So I think it's about what is the contribution of these hormonal changes, considering all these other things. And I don't know if we can answer that.
Yeah, a lot. Unfortunately, I'm not sure if I have the answer there either, but I would say, I mean, you would predict based on how, like Celeste said, there are certain taxa that perform these functions, the beta-glucuronidase or the sulfatases, It's not widespread necessarily. There are some on the bile acid side, some Clostridium scindens and other bacteria that and others, some other Clostridia as well that are key players there. But it depends on the gene. The GUS gene is primarily firmicute, bacteria within the firmicute phyla. The BG gene is kind of split across Bacteroidetes and firmicutes. So there's members of those taxa. So it's certainly, I think the microbiota that are present will definitely dictate the recycling aspect of it. So I would predict, I guess there would be a host effect there because of that. Like Celeste just said, there's really a lack of just such a lack of research in this area, whether it's the sex steroid hormones or there's more work being done in bile acids, but still at the molecular level, understanding the microbes involved, what genes are involved, and then the activity of those enzymes, it's all kind of slow moving. There's not a lot of people working in the area. And so Celeste we're trying to go that direction. And we had, Dr. Ridlan was on our committee, who's a really well-known bile acid expert, and he's down in the trenches there looking at the machinery that's metabolizing these things. But when you get into it, another thing to think about for those that are interested in this area, if you just say I want to look at the GUS gene, that beta-glucuronidase, and design primers to see the abundance of the gene. Well, that's not that simple either, because there's polymorphisms within the gene. So developing, it's not just one primer, it would be many primers depending on the taxa that you're interested in. And so so I think certainly the microbiota will have an effect here, but again, there's not much data to say this is how, and this is percentage of an effect. I do think, again, the microbiota are one player. I do think, of course, we want to look at it from their perspective, I think, of what they're responding to. But I do think, for the most part, we control what we put into our mouth. We control, hormones. I think it's more so the host is dictating what the microbes are probably responding to. Not that they don't influence us, but from the hormonal side, they're affecting the recycling. But I still think there's a lot of other signals and hormonal changes going on that I think are driving it. But certainly they're a player there that we have to think about.
I think it's just to add to that, if we're talking about does the initial composition influence things? I mean, even I think about it, I would assume so, because if you think about it just from a dietary fiber intervention standpoint, we look at responders versus non-responders. If you're looking for, someone's microbiome that's already well equipped to handle large amounts of dietary fiber, it's really hard to shift that microbiome. So I'm sure, the same thing probably goes for hormones, where depending on what your microbiome composition is to start, changes in that hormonal status may not be as robust in some people versus others.
Mm-hmm. And I think that's where I guess to follow up on that too, some of the germ-free mice data kind of shows that, I think, as well, that what they're inoculated with could certainly could affect that. But more work needs to be done, certainly.
Awesome. All right. Of course, there are more questions, but we're just about out of time. So that concludes our webinar for today. And we really wanted to thank Dr. Swanson and Celeste, soon to be Dr. Alexander any minute, for sharing your work with us today. And just to remind everybody to visit our website to register for the next webinar, which will be next Friday. And then we will hear from Dr. Yanina Pepino and her student Clara Salome about low-calorie sweeteners. So thanks, everyone, and we hope you have a great afternoon.